All right, so this exam, we're going to really focus on animal diversity and who are the animals and what makes an animal, and what can we say about the phylogeny of animals. And so, of course, this is based on features of animals and kind of the important characteristics. Um, so when we look at a phylogeny of animals, we have to kind of know what is an animal. Um, Animals have muscle tissue, they have nerve tissue. No other group of organisms have muscles or nerve tissues. Animals have, uh, they're multicellular, made of eukaryotic cells. They have organs and tissues. Um, and that's kind of a unique thing about them. Some animals straddle the border of what is an animal and what is not an animal. And even animals that are clearly animal may not look like an animal. They may sometimes look much more like a plant. Um, when we think of animals, we should think of things like symmetry, whether it's bilaterally symmetrical or radial symmetry. Most animals have a body cavity and internal hollow space. Animals start out as single celled organisms and so have to develop into multicellular organisms, and they do that through the accumulation of germ layers and features like segmentation, development of a gut, development of a coelom. Okay, and so here we have pictures of radial symmetry and bilateral symmetry. Here we have showing the two general patterns of development. Um, we have a group of organisms called the protostomes. For the most part, these show a spiral cleavage where the cells kind of twist as they divide and the tiers wind up kind of offset from one another. These cells in early development are in the first group have a fate that's already been determined. They are called determinate, and if you take one cell away, these organisms become defective. They have a tendency to form first their mouth, okay, by an invagination of cells that will um, produce the gut, and they tend to develop a coelom through a split in a pocket of mesoderm that develops near the near where the mouth is. The other type of development is the deuterostome development, and in that case we have radial cleavage where the tiers of cells lined up stacked on top of one another, and they're indeterminate. If you take one or two cells away early in development, you can wind up with identical twins, not defective embryos. They too go through an invagination to form the intestine, but instead of the first opening forming the mouth, it forms the anus, and the developing body cavity and mesoderm develops from outpocketings of the gut that happen kind of late in development and will determine form a coelom. Okay, so these are the great patterns. Some organisms, almost all organisms, have some sort of internal space. In some cases, it's a pseudocoelom. In other cases, it's a true coelom. But there are a few acoelomic organisms like flatworms. Um, and so they're acoelomate. We didn't talk about them, but you do see them. And looking early at development is a very important part for us because we can see lots of things. We can't see that for fossil organisms, but we can often identify other features that tell us where they are. Looking at the big groups of organisms, the ones that are probably most clearly not like most of the other animals are the sponges. They are sometimes referred to as parazoa. They don't have true symmetry, although it's sometimes present as radial symmetry, or it looks kind of like radial symmetry. And they don't have true organs or tissues. They're truly multicellular. They have cells that are different types, but they don't form organs or tissues. They don't stay close to one another. In this case, they have several types of cells, and one type, the choanocyte or collar cells, does most of the work. It traps food and passes it to other organisms. These guys do have kind of structural elements, kind of like connective tissue. They form internal structures called spicules. Here's a cross section of a sponge where you can see the internal cavity or spongiceal. They have pores because they suck water through their body walls. They create the choanocyte creates a current by having a beating flagella, and it traps food particles on its collar. So here you see some distinct cell types: the choanocyte, the amoebocyte, the porocyte, or kind of an epidermal type cell. There's a layer, kind of the mesohyle, that forms a middle layer between an inner and outer cell. But they're clearly different. Clearly, no muscle, no nervous tissue, no true tissues. An unusual structure. Okay. I didn't talk much about Ctenophora, but sometimes they're referred to as the Parazoa group. It's a small group of radial symmetrical organisms. The other really big group of radial symmetrical organisms are the Cnidaria. They're the jellyfish, hydroids, corals, and sea anemones, and they contain stinging cells. Cnidaria have a unique stinging cell, the nematocyst, and within, or nematocyst is the organelle, and then nematocyte or cnidocyte is the stinging cell. These have a little trigger and can inject poison into other organisms. So they carry a venom and can be used to attack prey, which are then pulled towards a mouth by kind of muscular tentacles. A unique feature of this organism is that they have two basic body forms: the free swimming medusa form, which is kind of an umbrella that beats and pumps, and then an attached form called the polyp. These guys have an internal blind ending gut. They don't have a gut that goes all the way through, and they really are only two layers of tissue: the diploblastic organization with kind of a disorganized mesoglial between the endodermis and ectodermis. Then the big group of protostomes are a bunch of animal phyla that form the mouth first, and we divide these into two kind of supergroups. The first of these is the Lophotrochozoa, which contains hundreds of thousands of species in many animal phylum. They're grouped together based on their genetics and that they have spiral cleavage, and many of them are going to form a trochophore, a feeding structure that's kind of a horseshoe-shaped ring of tentacles that traps particles, or they may have a Loaf of Four, which is a larval form that has two bands of cilia and will settle down somewhere to develop into an adult animal. So there's the Loaf of Four phyla, and then the Trochozoa phyla, which are the ones we focused on: Mollusca and Annelida. The Mollusca, really nice, and I really like the neat phyla in there, or classes in there, particularly the Cephalopods, maybe the most advanced invertebrates, large and complex. They have a foot that's divided into tentacles and arms so that they can trap prey. They have a visceral mass where their organs are and are surrounded by a mantle cavity that is used to usually ventilate a pair of gills and for locomotion. And so here we see that's a cuttlefish there and a squid at the bottom. So really complex animals, large, good organ systems, unique structures like a beak and an ink gland and a large brain, and unique skin that contain chromatophores and iridophores so that they can produce colored and flashing patterns. Okay, the other big group here of the protostomes are the Ecdysozoa. These are successful dominant groups that contain millions of species and trillions of individual animals. They too are protostomes, but more often exhibit radial cleavage than spiral cleavage, but they still have determinate cleavage. Their unique feature is that they have this outer cuticle that occurs in several layers and includes chitin, a complex polysaccharide with some proteins in it. And this forms an external skeleton that must be shed when they grow. So the process of ecdysoa. They lack cilia as a group and have amoeboid sperm, so they're kind of interesting that way. These Ecdysolines weren't nearly recognized as a group until we started getting DNA and RNA data, and then the molecular evidence became overwhelming that they're closely related, and that these phyla form a clade that's pretty unique. The largest group here is, of course, the arthropods, containing 2/3 or more of all animal species and the majority of animal biomass on Earth, common in all habitats. And here the cuticle forms an exoskeleton that has jointed appendages, and these are adaptable, can be adapted for swimming, walking, flying, and all sorts of different mouth parts. And so that's a crustacean you see in this picture. The next picture are the Hexapoda, the insects. A very successful group with wings as well as walking legs and feeding mouth parts and things like that. They have an open circulatory system, but they have a really nicely well-developed excretory system and are very adaptable, have a good nervous system with ventral ganglia with compound eyes, complex behavior, and then a respiratory system of tracheal tubes and these spiracles that actually pump air to the inside of the body. In marine forms, they will use gills or book lungs or something like that, but very advanced phylum. Okay, then when we talk about the deuterostomes, we wind up with the phylum Echinodemorata. This is a large group of organisms that show pentaradial symmetry as adults, but larvae are bilateral symmetry, and they have an endoskeleton of calcareous plates inside, kind of just below the skin, and a unique water vascular system. As adults, these are the Seastars and starfish. They have no discernible head as adults, but still have fairly complex behavior and are very effective predators often, even without a head. This water vascular system is kind of a hydraulic system that uses seawater that's pumped into these tube feet to give them lots of power. And so they have thousands of these tube feet, and they can use them to move slowly over substrates or to use that to open shells or attack other organisms. The other big group of deuterostomes are the chordates. And chordates, during their lifespan, all share 4 features. They share these pharyngeal slits that are used for feeding or respiration, an internal skeletal rod called the notochord. That's a long, flexible rod that's used for internal support. And then dorsal to the notochord is a hollow nerve cord that may be developed anteriorly to form a brain, and then a post-anal tail that contains notochord. Okay, there are some small groups here like Amphioxus, the cephalochordates, and the urochordates, but the major group here are the vertebrates, a subphylum where the notochord is supplemented or replaced by vertebrae, and these individual jointed discs then may have some notochord persisting between them. These guys develop an internal skeleton of cartilage or bone then with additional elements to protect organs and maybe have jointed appendages. And they have well-developed organ systems and can turn out to be very large, complex organisms. Okay, we talked about the fish, the different groups of cartilaginous fish, the ignatha, the chondrichthyes, and then the bony fish, the osteichthyes, and the subgroup Sarcopterygian, which gave rise to amphibians, which then gave rise to reptiles and birds and things like that. We are one of these groups of organisms that branched off the tetrapods. We've modified an amniotic egg so that we can have internal development, and we are a group of mammals called the primates that have grasping fingers and toes, binocular visions, and clearly have uh, not too distant relatives that lived in forests and climbed trees. Okay, but we as a group belong to a specific branch of these primates. We belong to the hominoids, which have an even better grasping hand with an opposable thumb, and instead of claws, we have fingernails. We lack an external tail, have an extremely large brain with adaptable behavior. We live in social groups, having long lives and slow development. Okay. There are several fossil species that provide intermediates between um, how humans are and living other primates, like chimpanzees, our closest relative. These fossil primates include species that are almost identical to humans, in particular Neanderthals, which lived between 300,000 and 25,000 years ago, had extremely large brain cases, as large as us or even larger, walked on two legs, used complex stone tool, fire, shelter, and even had a culture. So we can place ourselves right in with the group of animals. Okay, let's stop there. The exam will be similar to our previous exams. We'll have two essay questions for us to look at. We'll have about 40 questions, and we'll do that online in a 1 hour gap. It'll open up on Wednesday, close on Saturday. Um, Watch for some additional materials I'll put online there for some questions, and email me if you have any questions.